Hello anatomy students. This is part 2 of our PowerPoint, Hormones and Their Actions. What you need to know is how to describe classes of hormones and how they vary in their effects about cells. You should be able to detail how second messengers work to affect metabolic change in cells, explain the phenomenon of signal amplification and what that means for hormone concentrations in the blood, contrast upregulation and downregulation, describe and give examples of 3 types of hormone interactions, and explain the relationship between a hormone's half-life and its metabolic clearance rate. Okay, let's get started. So you know that hormones get deposited into the blood from different endocrine glands, and they are going to travel throughout the body, but they only affect cells that have receptors that are specific for them. So the cell receptors act as switches. Receptors for hormones are typically proteins or even glycoproteins, which basically means that you have a carbohydrate with a protein chain attached to it. These are found on the surfaces of cells, and they turn on and sometimes turn off metabolic pathways. But the biggest thing to remember is that receptors are specific for certain hormones. And as you can see in the picture, my yellow cell is a target cell for hormone A because it only has the receptor for hormone A, which is a circle. And the green cell is a target cell only for hormone B because it's got only the Y-shaped receptor on its cell surface, and hormone B is a triangle. But my blue cell in the center has receptors for both hormones A and B, so those cells are going to adjust their metabolic activities depending upon which hormone binds to the receptors at a specific time. This graph is showing us that with not much hormone concentration in the blood, you're going to get about 50% of all receptors being occupied. The straight line that you have up here at the top is showing us the total number of receptors, and you can see that the hormone receptor concentration starts to level off pretty early in the curve. And it's never going to quite reach 100%, but it can get pretty close. So, even with a low hormone concentration in the blood, you're going to get about 50% of the receptors being occupied. Now, steroid hormones, which are hormones that have a steroid backbone, the one that has the 5-ring structure, is going to directly affect target cell physiology, which basically means that these hormones are going to cross the cell membrane, enter the cytoplasm, bind to a receptor, and then together the hormone and its receptor, also known as the hormone receptor complex, they're going to enter the nucleus and bind to DNA, and it's going to activate mRNA transcription. And that's how it's going to change cell metabolism. It's going to directly affect transcription and translation in order to cause the cell to create different proteins. Now, protein hormones, peptides, and catecholamines, and remember that the catecholamines are epinephrine and norepinephrine, they can't enter the target cell. The reason why is because they're hydrophilic and they can't cross that phospholipid bilayer. So their receptors are found on the surfaces of cells, and they're going to utilize a mechanism called the second messenger. And I'll show you what's going on with the second messenger on the next slide, but you should know that these hormones are going to cause change faster than steroid action because you don't have to wait for transcription and translation to occur. An important thing to note is that not all hormones use the same second messenger, but also you should note that one hormone can utilize multiple second messengers. And then I just wanted to call your attention to the picture that you have on this slide. You can see that I've highlighted in yellow glow that the hydrophilic hormone, the peptide or the catecholamine, is going to bind to a receptor that's found on the surface of the cell membrane, whereas the hydrophobic hormones, these are the ones that are steroids, they actually cross the phospholipid bilayer, they enter the cell. In the cell's cytosol is where they meet up with their specific receptor, and then together they travel into the nucleus in order to affect transcription and translation. So second messengers are going to involve an enzyme called protein kinase, or PK. Protein kinases are going to change cell physiology, and the way that they work is they remove a phosphate group from ATP. So ATP is adenosine triphosphate, and that means that they're going to take one of the 3 phosphates off and then stick it onto an inactive protein or an inactive enzyme. Various second messengers are going to activate this protein kinase. And so hormones are going to stimulate this reaction to occur. So let's take a look at the picture that we have here. So you can see that in the yellow box I have inactive enzyme. That's my little protein there that looks like a little egg. And protein kinase then is going to take ATP and change it to ADP. And it does that by hydrolyzing our ATP and taking one of those phosphate groups and sticking it onto the protein. We call this now a phosphorylated protein, and that is now an active enzyme. So that's the way that our second messengers are going to work. But the only way that second messengers can do this is if a hormone, a protein hormone or a catecholamine hormone, binds to its superficial receptor on the cell membrane. So you do not have to know by heart all of the steps of how catecholamines use CAMP, but I just want to show this to you so that you can see all the different steps that are involved. And so what we have here is we have a hormone, our little red triangle, that is binding to our green receptor that is embedded in the cell membrane. And then this little pink circle here, this is called the G protein. So here's what's going on. So a hormone receptor binding is going to activate our G protein, which activates adenylate cyclase. And what adenylate cyclase does is it's going to convert ATP to cyclic AMP. So it's taking off one of the phosphate groups from ATP and then forming a ring. That's what the cyclic, that's what the little C means in CAMP. And so it's forming a ringed molecule. CAMP is going to activate our protein kinase. And the protein kinase, using the mechanism that I showed you before, is going to take that phosphate group and stick it onto an enzyme. So it's going to activate the enzyme by phosphorylating the enzyme. And that's going to cause various metabolic effects. And notice that this is plural, but we got here to the red star when only one hormone was attracted to one receptor. So we have very little hormone, but a much greater effect. And that's an important piece here, because that's what is happening in this reaction cascade. So we have a small stimulus where we have very little hormone that binds to a few receptors on the cell surface, but by using the second messenger system, you get a very great effect. So small stimulus, great effect. And what that means is that the cell doesn't have to expend energy and resources in order to make a lot of receptors on the cell surface. So one hormone molecule produces a multitude of enzyme molecules. And this is called the reaction cascade. Signal amplification. All right, let's talk now about upregulation and downregulation. So the number of receptors is going to determine how sensitive a cell is to the amount of hormone that there is in the blood. So with upregulation, we have our cells increase the number of receptors. And when you increase the number of receptors for a hormone on a cell surface, that means that the cell is more sensitive, or it's more reactive to the hormone that is surrounding it. So if we take a look at this picture, we have three cell membrane receptors, and then the little blue dots, those are the little hormones that are there. And we have low receptor density here. This is going to elicit a very weak response from the cell. But when these little blue hormone molecules bind to their specific receptors, look what happens. They are going to cause a metabolic change in the cell, which causes the cell to increase the amount of receptors on their cell surface. And because you have more receptors, the hormones are going to have more binding sites, and that's going to produce a very strong response to either the same hormone itself, or in my example that I have here, it could be from a hormone that comes later. So for instance, in the female sex cycle, estrogen is released in great amounts during the first 14 days of the female sex cycle, which means that the number of receptors for progesterone are starting to increase in concentration on cell surfaces. Progesterone is released after ovulation. But what this means is that the cells are primed. They're ready for the release of progesterone. And so just a little bit of progesterone is going to cause a great amount of response from the cells. And that's a really important thing in the female sex cycle, because if that egg that was ovulated becomes fertilized, the uterus needs to be prepared to nourish and grow that new embryo. And so by upregulation, you are causing these cells to create more glycogen, which is going to nourish the growing embryo in the case of pregnancy. And so in this example, hormone number 1 causes upregulation of receptors for a hormone that's going to follow later in time. So this is an example of upregulation. And downregulation, I'm sure you can figure out that that's going to be exactly the opposite, where you start off with a high density of hormone receptors on the cellular surface, but then as more hormone binds to it, it causes a different metabolic change in the cells, where they are going to diminish the number of hormone receptors, thereby causing a lowered effect or a lowered sensitivity to that hormone. So as you can see in our picture here, we have a high density of purple receptors on the surface of the cell. So at this point, we're getting a strong response, but that response is going to cause the cell to either suck in some of those receptors and degrade them. And so now we have a decreased receptor density. And so now, regardless of how much hormone there is surrounding the cell, you're going to get a much lower response, a weaker response. And this is what happens in people who have diabetes with adipocytes and insulin. So when they eat, typically insulin is released from the pancreas in order to stimulate cells to take up that glucose that's now in their blood. But if blood glucose levels are constantly high and the pancreas constantly secretes insulin, certain cells in the body, namely fat cells or adipocytes, are going to kind of like, quote, get used to the high amount of insulin in the blood. And so that's going to cause the adipocytes to degrade the insulin receptors, and therefore they're not going to respond to insulin as well. What that means is that the insulin does not stimulate the adipocytes to take up glucose molecules as it did in the past, and the person has elevated blood glucose levels all of the time, and that's exactly what diabetes is. So this is an example of down regulation. You guys can watch this video here on your own to see what happens with down regulation and how it plays a role in opioid tolerance. And now I wanted to talk about the three relationships that exist between hormones. The first kind is called synergistic. And what this means is that two hormones act together to improve the overall effectiveness of these hormones. And that's what you can see in the graph that I have in the bottom right-hand corner here. So hormone number 1, hormone number 2, on their own, they are mildly effective, but then you put them together and look at how high that bar goes. The effectiveness is going to go right up. FSH and LH and testosterone, they're all going to work together. They have a synergistic relationship in order to produce sperm in the testes. In a permissive relationship, one hormone is going to enhance cellular response to a second hormone that's going to follow later. We talked about this already with estrogen and progesterone, and here's how it works. So we've got estrogen represented by red triangles. During the first half of the female sex cycle, it's going to enter target cells, and it's going to stimulate a metabolic change in the target cell to develop more progesterone receptors on the cell surface. After ovulation, progesterone is going to be secreted by that empty follicle, and the progesterone now has places to bind, and that's going to cause the target cell to create more glycogen in order to prepare the uterus to support a growing embryo should fertilization occur. And then the last one is the relationship between insulin and glucagon. Insulin acts to lower blood glucose levels, and glucagon does the opposite. It raises blood glucose levels. And together, even though they're acting oppositely, they're going to help keep blood glucose levels within a very narrow normal range. So that's an antagonistic relationship. Hormones are going to be cleared from the blood and from around cells after the purpose is served. This is going to happen at target cells where they can degrade those hormones. The liver is going to help by excreting hormones in the bile, and by the kidney by excreting hormones in the urine. Back in our previous presentation, I mentioned that steroid hormones are going to have a longer half-life because they're bound to transport hormones in order to help them through that watery environment of the blood, whereas catecholamines and peptide hormones have a lower half-life because they are water soluble, and so they are more exposed to enzymes and they will be degraded a lot faster. The other, and by hormones being cleared, what I mean here by target cells is that the hormones are going to exit the blood and bind to the target cells, and then they can be removed by enzymes from there. And so this graph is just showing you that the half-life of lipid-soluble hormones, and you've got time on your x-axis, and then concentration should be on the y-axis here, but you can see that over time the lipid-soluble hormones are going to remain in the blood for a longer period of time at more constant levels than our water-soluble hormones, because again, they don't bind to our transport proteins, and therefore they are exposed to the enzymes. So the lipid-soluble ones have a longer half-life than the water-soluble hormones. Okay, so number 1, which classes of hormones affect cells indirectly? What I want to know is which class of hormones uses the second messenger system? These would be the hydrophilic hormones, peptides, monoamines, the catecholamines. All right, question 2. Are the hormones listed above hydrophobic or hydrophilic? Whoops, if you were listening carefully, I gave you the answer to this. They are hydrophilic, exactly. So remember that to use the second messenger system, the receptor has to be on the surface of the cell because these hydrophilic hormones can't cross the phospholipid bilayer. All right, one hormone raises blood glucose and the other one decreases blood glucose. Which term describes the hormones' relationship? If you said that they are antagonists, you would be correct. All right, anatomy students, thanks for watching this video. I hope you learned something and I hope you enjoyed it. Talk in class. Bye.